Hello, my name is Emily Leonard, and I am a junior biology and public health double major at UMass Amherst. I am a research assistant in Alicia Tamilergi's lab, and this presentation will be about our current meta-analysis of single-cell sequencing of human pancreas beta and alpha cells. I worked closely with Emily Marks and Emily Formato and Wen Lang. So I want to give you guys a bit of background information on the endocrine pancreas before we get started. The endocrine pancreas is composed of numerous isolates that contain 5 different endocrine cell types: alpha, beta, delta, epsilon, and upsilon. The beta cell of the isolate secrete the hormone insulin, and the alpha cells secrete glucagon. Both hormones are critical for blood glucose homeostasis, which, as I will mention, is a main cause of developing diabetes. So let's get to it. There are 2 types of the disease diabetes mellitus. Type 1 diabetes is a chronic autoimmune disease characterized by insulin deficiency, and the immune system of individuals with type 1 diabetes actually attacks pancreatic beta cells and disrupts their ability to produce insulin. Without the ability to produce insulin, glucose accumulates in the bloodstream and can cause serious and potentially life-threatening complications. The exact mechanism for why the immune system attacks the isolate beta cells is unknown. However, it is theorized that there is a genetic alteration or an environmental exposure causing the development of the disease. Type 2 diabetes, on the other hand, is a chronic disease characterized by insulin resistance and severe beta cell dysfunction. The body resists the normal effects of insulin and fails to drive the uptake of glucose. As a result, the pancreas increases its insulin production to help glucose enter the beta cells. However, it eventually becomes overworked and blood glucose levels are increased, again causing serious and potentially life-threatening complications. Type 2 diabetes is now considered a global pandemic affecting people in both developed and developing nations. In 2015, more than 415 million people were diagnosed with type 2 diabetes worldwide, and this number is expected to climb to 642 million by 2040. Similar to type 1 diabetes, the exact mechanism by which it arises is unknown. However, there is evidence that a heritable genetic component and a strong life factor component are part of the development of this disease. People with both type 1 and type 2 diabetes are at higher risk for comorbidities such as heart and artery disease, hypoglycemia, ketoacidosis. And type 2 diabetes individuals are actually at a higher risk for developing certain cancers as well, such as bladder cancer, breast cancer, liver cancer, and colorectal cancer. And so the pancreatic isolate is characterized as having the lowest levels of intrinsic antioxidant defense, and therefore when reactive oxygen species, RSOs, accumulate in the body for prolonged periods of time, the isolates, especially the beta cells, are at greater risk for chronic oxidative stress and adverse effects, which could be leading to the development of type 1 and type 2 diabetes. However, in order to make this conclusion, we must gain an understanding of the antioxidant defense genes in the cell types of the pancreas and how these changes can lead to the effects, lead to the different types of diabetes. And that's where our meta-analysis comes in. So specifically, we looked at RNA sequencing data, and RNA sequencing data is a way to compare the genomes of normal individuals with individuals of type 1 and type 2 diabetes. With RNA sequencing technology, we're able to quantify the transcriptome of individuals, which is the complete set of transcripts in a cell and their quantity. By understanding the transcriptome, we can interpret the functional elements of the gene and quantify the changes in gene expression levels of each transcript under different conditions, thus allowing for more insight into the development of a disease. We specifically selected 5 studies based on a PubMed search for single-cell RNA sequencing of pancreatic isolate cells. All studies had to contain publicly available single-cell transcriptomic sequencing data for both normal and diabetic human pancreas donors. The studies also needed to provide count data and properly randomized trials. Once we selected the studies, we downloaded the single-cell RNA sequencing data from NCBI Gene Expression Ombuds, and each of us took on the role of annotating the cell type for one of the studies. All sequence cells were classified based on key marker genes, which included key hormone genes such as glucagon, which is found in the alpha cells, and insulin, which is found in the beta cells, somatostatin, gherkin, and ovastatin prepopeptide, and pancreatic polypeptide. We also assessed genes that encoded ACNAR cells, such as serotine protease 1 and pancreatic lipase, as well as genes associated with ductal cells such as keratin 19, secreted phosphoprotein 1, and hepocycle nuclear factor 1 beta. We chose these key marker genes because many of the studies we looked at also used them as a way to annotate their cell types. After a crude classification on key marker genes, each cell type was rendered in a violin plot using JMP Pro software. If a cell expressed multiple key gene markers or was considered an outlier, it was excluded. After that, we combined the data sets. However, this was a bit challenging because the papers were not uniformly normalized in their data. So we had to convert counts per million values to reads per kilobase million, and then to transcripts per kilobase million to normalize the units. And with such a large amount of data, it was very difficult and time consuming to actually normalize the data, which led to us having difficulty getting our data analysis done. However, once the data sets were combined, we generated a fold change value and a p-value for each gene using an ANOVA statistical test. So here you can see the violin plots that I actually created for the paper I looked at, Euron et al. 2016. And as you can see here, the key marker genes in cells characterized as beta cells was very high in the insulin key marker gene. And similarly, in cells characterized as alpha cells, they had high expressions of the glucagon gene, which makes sense because the beta cell secretes insulin and the alpha cell secretes glucagon. Okay. So here you can see our top 10 differentially expressed genes in the beta cells. Here they are. You can see the gene ID, gene name, the function, the fold change, and the p-value, and the NCBI entry number. And while we were unable to do a complete data analysis, we used David's software to determine the pathways that were associated with these upregulated genes. And we were able to determine that 4 out of the top 10 differentially expressed genes in the beta cells were involved in the VEGF-A, VEGFR2 pathway. And this pathway specifically activates angliogenesis, leading to proliferation, survival, sprouting, and migration of endothelial cells. And this pathway has been found to be related to cancer, which is interesting because people with type 2 diabetes are at a higher risk for cancer. And additionally, we were also able to determine through David that 4 out of the 10 genes are involved in ubiquitin-like modifier processing, activation, and conjugation or deconjugation. Like I mentioned previously, we need to complete more data analysis in order to draw conclusions about the mechanism by which type 1 and type 2 diabetes arise. But for now, this is what we have for our data to share with you guys. We hope this week to get into more analysis using Ingenuity pathway analysis software to obtain a z-value for each gene in order to assess how much it is upregulated or downregulated. So further directions that we have for our research is that once we have completed our data analysis, we hope to determine differences in antioxidant genes between normal and diabetic alpha cells, in addition to the beta cells that we've already looked at. We also hope to further characterize beta cells by NRF2 related genes to determine dysfunction and a connection to oxidative stress. Additionally, we hope to compare the mechanisms by which type 1 and type 2 diabetes arise to changes seen with exposure to environmental toxicants, such as PFAS. And our results will then be able to be used to identify potential risks to human health, which was our main goal. So, thank you so much for taking the time to listen to my presentation, and please feel free to contact me if you have any questions about our current research, our directions moving forward, or any of the complications we had during our meta-analysis. Thank you.